Hi everyone, my name is Hans-Peter and I'm a study director at ViruSure. Today I will present you a case study of our identification of a viral contamination in a biopharmaceutical product using nanopore sequencing. First of all, I want to give a little introduction of what our company does and who we are. So ViruSure is a GMP and GLP certified CRO based in Vienna, Austria, and we are part of the Asahi Kasei group. We are offering a wide variety of biosafety testing services to the biopharmaceutical industry. The tests include in vivo and in vitro adventitious agent testing, as well as real-time PCR testing for the detection of contaminating agents, virus clearance studies to demonstrate virus reduction capabilities in the manufacturing process, genetic stability testing to observe the stability of production cell lines, virus stocks over the complete production process, and many more, which you can look up on our website if you're interested. So let's get into our case study where we identify the contaminant in one of our customer samples. For the detection of the contamination, we use a cell culture-based assay called in vitro adventitious agent test. This test is used for the detection of viruses in a variety of biological samples, like recombinant proteins, monoclonal antibodies, and cell and gene therapy products. To ensure a broad range of detection, at least 3 different cell lines are used to maximize the chance of detecting a contaminating virus. These cell lines should contain 1 cell line of human origin, 1 of primate origin, and 1 of the same species as the test item itself. For the evaluation, 3 different endpoints are used. One is the cytopathic effect, where we observe cell health over the time of the experiment. Haemorrhagic, where we add erythrocytes that are attaching to virus-infected cells. Hemagglutination, where we observe for cell clumping due to surface antibodies attaching to the viruses. In our case, the sample was a bulk harvest sample from a bioreactor, where cells of hamster origin producing a recombinant protein were present. So we inoculated virus cells with the test sample, and 4 days after inoculation, we could see a clear cytopathic effect when comparing the test sample cells with the negative control cells. In such a case, as our product should always be clear of viruses, we have to perform an out of specification or OS investigation to evaluate every potential factor that can have an influence on the outcome of the experiment. This includes check of the controls, equipment, possible cross-contamination from positive controls, operator errors during the handling and preparation of the test sample, and many more. After this investigation, there was no indication of errors from our side that could have an impact on the results in a way that this turned out positive, false positive. Therefore, we had to identify the contaminant, and therefore we had different options available. One was cell culture based, but these are long and drawn out investigations with limited specificity. Others would be qPCR antibody assays. These have high specificity, but the list of possible PCRs and antibodies is very long, so it's more a game of chance if you actually detect the virus or not. The third one would have been transmission electron microscopy. This could reveal the structure of the virus and give a hint on its identity, but it also only has very low sensitivities. So we decided to go on with nanopore sequencing, as we had recently just acquired a MinION device, and for this approach we would not need any upfront information for the identification of the contaminant at all. So we started by doing separate RNA and DNA extractions, used different kits for library preparation, and then ran all our samples on a MinION flow cell. For evaluation, we used the epitome WIMP workflow, and for possible contaminants, we tried to confirm them by mapping against the reference genome and do a second confirmation and title determination using real-time PCR. First, we did start with our DNA extraction. We only had a very low amount of input DNA, so we decided to use a kit including PCR amplification. In this case, we used the PCR barcoding kit and performed the run on a MinION R9 flow cell for 72 hours. We generated around 13 million reads, and these reads were analyzed with epitome WIMP, and most hits that we obtained were of retroviral origin. These, however, were present in the test sample as well as the negative control, so the most likely origin are endogenous retrovirus from the virus cells, and the contaminant was therefore not introduced by the test sample itself. So we went on with the RNA extraction. Our first runs did not yield any good quality data, so we did some troubleshooting. We checked the quality of the flow cells and also the extraction recovery, but all the flow cell checks and also the qubit measurements looked okay, and all our repeated runs did not yield any more and better data. However, as you can see on the table on the side, that we still obtained a few reads that were associated with the epizootic haemorrhagic disease virus. These hits were only present in our test sample and not in the negative control. Therefore, we thought that maybe the reverse transcription was performing poorly. As RT is highly efficient for single-stranded RNA, it lacks the capabilities to reverse transcribe double-stranded RNA. Therefore, we included a double-stranded RNA denaturation step before the actual reverse transcription, and this resulted in higher yields and a drastic increase in read counts for EHDV. So we used this data set and mapped it against an EHDV reference sequence. The EHDV genome is segmented, and we got good coverage for 8 of the 10 segments. Still, 2 of the segments were not covered at all, so we decided to do another confirmation by qPCR. We made 2 primer designs, one into the not detected segment and one in a detected segment, and compared the results. Both setups turned out positive and gave titers of around 10 to the 8th genome copies per milliliter. So what are the possible origins of the contamination? So EHDV, or in general reoviruses, are endemic in most parts of the world. EHDV and other reoviruses will therefore be a risk concern wherever bovine components are present in cell culture medium, for example, by the addition of bovine serum. As reoviruses have a high titer viremic phase, only one infected cow will already result in significant levels of contamination in pooled sample. To reduce the risk, it is necessary to carefully source and test your reagents, followed by inactivation, for example, by the usage of gamma-irradiated serum. So to sum it up, with an optimized protocol, Nanopore sequencing proved to be an effective tool for rapid identification of contaminants in the manufacturing process without any prior knowledge of the contaminant itself. Besides that, it shows additional potential for other applications within the field of biosafety testing services, such as sterility testing to test for the absence of microbial contaminations, as well as genetic characterization of production cell banks. Thank you for listening.